Good morning, everyone. This is the part 2 of the Tuesday discussion of the human microbiome. This is a figure from this paper down here, the 2012 Nature paper, which is called Structure and Function of the Healthy Human Microbiome. This is on the Camino page, and it's also in there's a link to it on the modules overview for week 8. It's also in the folder that you're going to use for the week 8 lab this afternoon. So you should all download this paper so that you can take a look at it. There's also a link here to a separate YouTube video on the Human Microbiome Project, right? So as the previous slides mentioned, there are 2 approaches that were used in this paper. So there's sort of cataloging the who's in the population using the 16S ribosomal RNA sequences, which you amplify by PCR. And then there's just basically sequencing all the DNA. So in the introduction to this, it talks about getting samples from a bunch of different sites shown here from, I think, 240 people. I believe, if I remember right, there was, let's open it here next to me, there was there was actually more men than women. So 129 men, 113 women. And they took samples from all these different spots, 15 sites in men and 18 in women. And they analyzed the microbiome in each of those samples by both of those methods, right? So the idea here is to get a fairly comprehensive picture of what's in the microbiome at each of these sites, and ideally from a metagenomic analysis, what is going on there as well. So I'm just going to hit some of the highlights here, and I want you to look at the paper yourself as part of the lab this week. So in this figure, this is actually figure, let's see, this is figure 3 in the paper, but I think it's fairly illustrative, so that's why I decided to show this for you. So on the left-hand side here in panel A is data from metagenome analysis. Then on the right side here is data from the 16S analysis. And you'll notice that this is at sort of the genus level, and this is at the individual species level. And here what we're looking at is some of the most common species in each of these locations. Now the size of the circle here is reflecting the relative abundance of that species in that niche. Okay, so the niche goes like this, the same species goes across here. And you see here, not surprisingly, that different species are more or less abundant, larger or smaller components of the population at that niche. For example, so just to go through these real quick, here we have basically the nose. This is the skin behind the ear. This is in the mouth, the buccal mucosa, that's basically on the lining of your cheek, the top of the tongue, the supragingival plaque, so that's plaque above the gum line. We also have over here subgingival plaque, so that's below the gum line on your teeth. This is a fecal sample as an indicator of what's in the colon, and then this is a vaginal sample. See over here, they have it broken down into a few more components, 3 different anatomical locations in the vagina. And then here, these are all in the mouth, at different places in the mouth. 2 different places on the skin. This is on the inside of your elbow, again, behind the ears, and then in your nose. So this is sort of an overview of the different sites that they sampled here. So again, you can see different species. Propionibacterium acnes is abundant in the skin and nose. This is associated with acne. This lactobacillus species is, and actually a couple other lactobacillus species here also very abundant in the vaginal sample. Some others that are abundant in on the skin here, Corynebacterium species, and this Prevotella is abundant in the colon, and in the mouth, the Streptococcus mitis, which is associated with dental cavities. Again, over here at the genus level, you can see Lactobacillus, very abundant in the vaginal samples, Propionibacterium in the skin, Streptococcus in the mouth, Bacteroides in the colon, and Corynebacterium staph and Moraxella, again in skin. Okay, so the point is here that different locations in the body have different species compositions and have different dominant species that are adapted to that location in the body. We're going to go through some of these in a little more detail, but of course I want you to take a look at this paper, and this is going to help with your lab this afternoon as well, which I'll go over in a few minutes. Down here, the difference between this one and this one is that this is looking at potential pathogens, in other words, bacteria that are associated with disease. Now, as they talk about in the paper, overt pathogens that don't have any normal function in the body are not common at all in this, but there are many opportunistic pathogens, and this is a concept we're going to talk about on Thursday, that are common in these different niches. So an opportunistic pathogen is a bacterium, usually, that can cause disease, but it's there as a normal part of the commensal microbiota. So it lives in association with humans, and it only acts as a pathogen when something changes. For example, when there's physical damage, as in a wound or a burn, or when there is compromised immune system. All right, so these are just showing a lot of these are essentially opportunistic pathogens. And it talks about this in the paper. All right, this figure is, let me see, this is figure 4 from the paper. And this is kind of connecting this to the genomics that we were talking about. Not this first part here. This is just showing the distribution of different Streptococcus species. And what this is getting at is variation between individuals. So each one of these lines is a sample from a different person. Now the paper, as you'll see, talks about 2 kinds of variation. One is what they call alpha diversity, which is the diversity of species within a niche. So how many different species are there? What's their relative abundance? There's a quantitative measure of that. Then the other type of diversity is beta diversity, the difference between people at the same niche, at the same anatomical site. How much diversity is that? And you see here, these are all from the top of the tongue, and they are, there's a lot of variation in the different streptococcal species that we see on the tongue here between these different individuals. In this one, the strep species make up over 60% of the entire population in that sample from the tongue. In this case, it makes up maybe 5%, right? And then there's different ranges of abundance of different species within that. This is looking at variation in the presence of different metabolic pathways or proteins systems in these different streptococcal species. You see some of them are pretty conserved. This is for uptake of glucose. Some of them are only rarely present. This is a different transport system for glucatol and sorbitol, right, which is only there in one, right? So there's, within the genus Streptococcus, there's a fair amount of variation in the ability to take up these nutrients. This is looking at genomic variation within one species, Streptococcus mitis, which is quite common in the mouth. That's the light blue one here. It's actually, it's not a huge component of the streptococci, but it's pretty consistently there. And what this is showing is the presence of different parts of the genome, some of which are there in virtually all um, of the isolates, and some of which are not very common at all. Now, this is a log function, so well, anyway, we won't go into that further. And then this is just looking at specifically these two genomic islands. If you remember from our discussion of genomics, a genomic island is a mobile element that has different genes in it that may be associated, for example, with pathogenesis. And that would be what we would call a pathogenesis associated island, right? Again, it doesn't matter the details of what these are, except to say that these are thought to be involved in virulence in this bacterium. All right, so these are two sort of complex figures from this paper. You don't need to study this paper in great detail. You don't need to know all of it, but I do want you to use it as sort of a starting point as you're going into your lab exercise this afternoon. All right, just a few more details, not details, but just a little more on each of these sort of niches that are talked about in the paper. The human skin is a very large surface of our body, exposed to all the outside world on an ongoing basis. And its different regions of skin are definitely quite distinct. We have oil glands on our skin that release oils onto the surface to keep our skin surface moist and flexible. These oils tend to have inhibitory action towards microbes. So a microbe that's adapted to live on skin would be able to handle and even metabolize these. This is one of the things that Propionibacterium acnes does, is it's quite good at consuming the oil secretions on their skin. You have hair follicles. The sebaceous glands and the sweat glands are actually generally associated with. Some parts of your body have lots of hair. Well, used to have lots of hair, but you know what I mean. And some parts of your body have very little hair, which again, so that's going to affect the amount of oil and sweat, etc., that are present on that part of the skin. We should say that sweat has a lot of salt in it. So the regions which get very sweaty, and when that sweat dries out, they have a high salt concentration, high salinity. So anything that's going to be able to live there is going to be able to have to tolerate that salinity, right? So these are some of the bacteria that typically are common in different parts of the skin. The relative abundances of these is going to vary depending on where you are and depending on who you are. A lot of these are gram-positive. Propionibacterium, Corynebacterium that were mentioned in the paper. Staphylococci, if you remember, Staph aureus and Staph epidermidis grow on mannitol salt agar, which has a high salt concentration, so they can tolerate that. And some lactobacillus species, although lactobacilli are definitely more common in other areas. Some gram-negatives are found on skin as well. Typically, though, it's a lot more gram-positives. And again, it really depends on where you look. This paper, this review article, was talking about the diversity of the human skin microbiome, and they aggregated data from a lot of different sources, looking at the composition of species in different locations, anatomical locations on skin. And so, if you're in anatomy right now, you might know more of these. It's like the belly button, and there's, it depends on whether it's sort of an open surface, how much hair is there, if it's a crease, if it's down in your toes, between the toes is going to tend to be much moister. And you see that all of these have different groups of bacteria. The actinobacteria are gram-positives. These bacteria are gram-negatives. The firmicutes are also gram-positives. And so again, different groups of bacteria depending on where you are on the body. I should also point out, as it wouldn't surprise you, that the amount of bacteria and the composition of the bacteria also depends on things like hygiene and climate and, you know, the clothes that people wear, things like that. So those can all definitely affect the skin microbiome. All right, moving inside the body now, the mouth is really sort of the second richest microbiome in the human body after the lower intestine. Not surprisingly, it's warm, it's moist. You know, skin can be drier and cooler, depending again on the climate and on where you're on the body. But the mouth is a more consistent environment, and it's typically pretty nutrient rich because it's the site, of course, that you're eating and drinking, and so you have, you know, the food that people are eating or whatever they're drinking that is at least passing through the mouth and leaving behind nutrients. There have been many hundreds of bacterial species detected in the mouth, mostly by a sort of population analysis. A lot of them can't be cultured, and you know, this is true of other niches in the body too, but it's especially true in the mouth for whatever reason that a lot of them have not been cultured yet. So as you saw from the paper that the Nature paper we talked about really briefly, there are lots of streptococci in the mouth. There are other bacteria, including obligate anaerobes. This is an interesting feature of the mouth, that there are many obligate anaerobes, and some of these cause significant health problems. So Fusobacterium and Porphyromonas gingivalis in particular is associated with periodontal disease. Streptococcus mutans is associated with dental cavities in the teeth, right? So if you want to go to dental school, then you undoubtedly will become familiar with the oral microbiome, because a number of members of that microbiome are associated with dental or oral disease and periodontal disease. So let's see. Having said that, though, we don't want our mouth to be sterile, just like we don't want our skin to be sterile, etc. And this is a point that I should have made more strongly. The healthy microbiome is actually both a normal part of our body and often important for the defenses of the body. So controlling the ability of pathogenic microbes to colonize. Going further into the oral microbiome here, we have sort of close-up views of teeth showing the biofilm, the plaque biofilm that grows on these teeth, which is where these bacteria like streptomutans and Porphyromonas and Fusobacteria live. This is showing the sort of the buildup of dental plaque. You have sort of non-microbial components that are comprised of things like saliva proteins, food particles, things like that, that the bacteria naturally grab onto because they are going to use these as sources of nutrients. And then as these bacteria are growing and proliferating, they start secreting their own polysaccharides to create this biofilm, and you get a complex community here. And the more this community builds up, the more problems it creates. You see that this community can induce inflammation in the gums down here, which over time can cause the gums to recede, exposing the roots of the teeth. The plaque here also, as these bacteria, these streptococci, if you remember, these streptococci are what we call lactic acid bacteria, because they secrete, they carry out fermentation. That's the only form of metabolism they have. And they secrete organic acids, and these organic acids start dissolving the enamel of the teeth and allowing the underlying structure to be damaged, which is how you get cavities. And these cavities can happen here on the outside, or they can happen here on the surfaces of the teeth and the sort of the grooves and creases on there. And so keeping this plaque under control is what you're doing when you're brushing your teeth or when you're going to the dentist for a periodic cleaning, which is really important to sort of scrape this away, because as it grows, it gets harder and harder for your toothbrush to actually pull it off. So this is or scrape it off. So at any rate, the oral microbiome is really important for oral health, and oral health is actually important for a lot of other things as well, although we won't talk about it right now. Right now, moving down the body as we go from the mouth down through the stomach, there's not much that normally lives in the stomach. We did and will talk a little bit more about Helicobacter, which is a bacterium that causes gastric ulcers, but really there's not much of an endogenous microbiome here. But as you get further and further down through the small intestines, and particularly into the large intestine, the microbial community grows. And you can see here there's some differences in depending on where you're looking in the gastrointestinal tract, but the highest density concentrations of up to 10 to the 13 CFUs per gram of solid contents in the colon being bacteria. So you have massive amounts of bacteria down here. Most of them are obligate anaerobes. I should point out, just to go back here for a second, again, some of these are obligate anaerobes too, like the fusobacteria, and they're able to live there because the formation of this biofilm present and in the context of the inside of your mouth prevents the diffusion of oxygen in there. So even obligate anaerobes can grow there. But down in your colon, the obligate anaerobes become the vastly dominant population down there. The Enterobacteriaceae is where E. coli fits in as a facultative anaerobe, but a lot of the rest of these are, most of the rest of these are obligate anaerobes, or in the case of the streptococci, are indifferent to oxygen. Okay, one other thing to point out here is that as you get away from the stomach, the pH goes back up closer to neutral by the time you get to the large intestine. All right, this is just an overview of some of the major components of the colon microbiome. Okay, again, there's more about this in the paper, and if you choose to do one of the papers on, fecal transplantation or probiotics to treat intestinal disease, you will encounter more of these species. All right. So what are the benefits of this relationship to humans? We have microbes everywhere in our body, and as we said, we have an awful lot of them in our intestines, so it's important to think about why we have so many. Are they just free riders that are extracting nutrients from the food that we're trying to digest, or are they actually doing something for us? And in fact, it does seem like they're doing a number of things for us. They are producing certain vitamins that our own cells can't produce that we then ingest. So these vitamins are not necessarily present in the food when we eat it, but are synthesized by the bacteria as the digested material is passing through our intestines. They actually break down some things that we can't break down very well ourselves. Insoluble polysaccharides that the bacteria are better at digesting than we are, including cellulose. And then those, at least some fraction of that material is typically released from those bacteria, usually as short chain fatty acids, which we then can absorb. And so we actually get a boost of the numbers I remember reading are like 10% in our energy yield from our digestion because of this help from bacteria. Of course, they're getting food themselves, so they're getting a benefit out of it as well. And then it's also been shown that proper immune function depends on our intestinal microbiome. they would seem to sort of prime the immune system to respond to pathogens when they're consumed, as we'll talk about on Thursday. You know, the fecal oral route of transmission of pathogens is often really important. And there's a number of other aspects of our immune system that seem to be tied to having a normal, healthy microbiome. One of the ways that this is looked at is by looking at the growth of what are called gnotobiotic animals. Those are gnotobiotic is spelled GNOTOBI. That equals germ-free. These are bacteria that, these are animals, usually mice, for example, that are raised in an environment where they don't encounter bacteria. So they're raised in a sterile environment, and they do have a number of problems in digestion and nutrition. So from that, we can infer that the presence of a healthy intestinal microbiome may be important for nutrient absorption in a lot of cases. Okay, so there are a number of benefits. This is a link to a TED Talk that you can watch on this. There are two other slides here that I want to show you to wrap up. One is on the question of probiotics. So probiotics are live microbes that, when they are ingested, may provide a beneficial effect to the human body. It's generally assumed, of course, that this is through its impact on the GI tract microbiome. Although, let's see, we didn't actually talk about the vaginal microbiome, I just realized, but if you choose to focus on that, there are probiotics that are thought to have beneficial effects on the genital urinary tract too, so they make their way there eventually. So essentially, these are often yogurt is an example of this. There are many other fermented foods that have live microbes in them, and they are thought to potentially be useful for restoring some kind of balance in again, the GI tract or the genital urinary tract. Often these are lactobacilli. And I would say kind of the jury is still out on a lot of these, but there have been studies showing, significant effects. And one of your options for lab this afternoon is to look at a paper on the role of probiotics in treating GI tract disease. So I think this is an interesting topic for discussion for sure. I'm sorry to not be spending much time on it. We can talk about it on Thursday if you like. But there's a lot of interest in sort of this external manipulation of the human microbiome through live cultures. Another thing that is in play in the lab this afternoon is the concept of fecal transplants. Again, this is for conditions where there's been a disruption or a dysbiosis, and usually the intestinal microbiome is what you're going to be dealing with when you're talking about fecal transplants. The most obvious example is Clostridium difficile, which is an obligately anaerobic pathogen that causes a very dangerous disease called colitis. Clostridium difficile is sort of a disease for the modern time, and that when you treat with oral antibiotics, something else, often you have collateral damage to your gut microbiome. Clostridium difficile is pretty resistant to antibiotics, so it can then overgrow and cause an intestinal disease as a result of an antibiotic treatment for something else. Okay, so the idea of a fecal transplant is essentially to take bacteria from a healthy person, transplant it into a person that's experiencing some kind of dysbiosis, including Clostridium difficile infection, and sort of help them restore their normal gut microbiome. So FMT stands for fecal microbiota transplantation. Not well regulated at the moment. And so there's a lot of sort of interest, both interest and debate about the future of this as a therapy. Again, this is one of your options for the lab this afternoon. Okay, I'm going to stop sharing this now and go to the show you. Sorry, give me just a second here. Well, I'll talk about it in class, what you need to do for the lab. Okay, so I'm going to wrap this up now and